All right. Hi, everyone. So, administrative stuff / question number 1. Here is the rest of our semester. You can see that I shifted around topics a little bit. When I go back to my office, will post the vaccines assignment. It's not posted yet, but it's easy enough to hit post. The adaptive immunity lab part for tomorrow is already there. At least I think the first half of the questions you could answer now, because there are things like, what did you put in this well? What do you predict will happen? And then there's like make a graph, and you kind of need the data for the graph, but the questions before the make a graph you could do right now. What's gonna end up happening is we're gonna set some stuff up, and I'm gonna have to email you the data, because the cells are gonna have to be in culture a little bit longer. So that'll be the plan on that. And then I'm doing vaccines on Fridays specifically because that assignment is up the following Monday. So this is all this information. And so there's one thing on here that is not listed, which is the lab report, the final lab report. I'm going to give you back your lab reports with comments tomorrow. And I'm going to put 2 things up here. RE the lab report. We'll say the lab report you can give me on Monday. But honestly, if you give me the lab report by this day, you don't get a late penalty. Late penalties do not start accruing until 5 p.m. on this day. Because I know what my schedule's like. I got plenty of other stuff to grade. That'll be the way that works. So you're going to get the graded thing back tomorrow. It'll be due on Monday. But as long as you give it to me by 5 p.m. next Friday, you're good. As you also might guess from looking at this information, I will send you some info about I'll give you a doodle poll next week for a review session for the final. Other questions, comments, concerns, anything else I'm forgetting? I'm trying to keep on top of it. It's questionable how I'm doing, but I'm trying. So today I'm going to say a little bit more about transplantation, and then I'm going to talk about therapies that are used for some of the different clinical conditions that we've talked about the past few class periods. Many of them are, of course, modifying immune responses because the clinical conditions are immune related. And so we can kind of think of them broadly as immunotherapies. But as I said, the first thing I need to do is finish up a little bit of transplantation related information. We spent quite a bit of time on Monday talking about solid organ transplantation, the importance of matching with solid organ transplantation, and kind of all of the details surrounding that. And so we're finished up talking about bone marrow transplantation and the specific issue of graft versus host disease, as well as the fact that we now tend to transfer just stem cells that have been purified instead of whole bone marrow to avoid graft versus host disease. But the one piece that I didn't get a chance to talk about with regard to bone marrow transplantation has to do with MHC matching. Because we talked about MHC matching with regard to solid organ transplantation. But there is an additional sort of complication when we think about MHC matching with bone marrow transplantation. In reality, we don't often have to match as many MHCs, so you actually can have less of a match for bone marrow transplantation and still make it work. But some of the issues with regard to bone marrow transplantation and matching are a little bit different. And in thinking about this, we need to remember a piece about what happens during bone marrow transplantation. So remember that what we're going to do is we're going to have our patient who is receiving a bone marrow transplant. This might be because they need to get radiation and chemo for something else, and we know that that's going to wipe out their immune system. Or it might be because they are having a problem with their immune system. Say they have SCID, and so their B cells or T cells don't work well. So we wipe out those ones that don't work well, and we put in new cells. In either case, we're sort of potentially wiping out what we have in terms of immune cells and transferring in hematopoietic stem cells into that individual. And so when we do that, we are reconstituting all parts of hematopoiesis. So in fact, that person we saw who was empty, who's getting kind of refilled, they were missing kind of everything on this slide. And we had to rebuild basically all these things. So they were missing, when we irradiated them, they started missing B cells and T cells and NK cells, and we regrew them some new B cells and T cells and NK cells. And they're now missing macrophages and dendritic cells and neutrophils and eosinophils and basophils and mast cells, but we regrew them some from their bone marrow. They will eventually have their red blood cells coming from this bone marrow as well, etc. So we're replacing all of these things. There are some cells, of course, in the body that we are not replacing. It's not like they're invisible. They still have like the structure of the body. It's just the immune cells, the rapidly dividing cells that went away. And some of those cells that are not killed will be, things like epithelial cells. So I'm going to draw out a little scenario on the board, and then we're going to think about what would happen in a bone marrow transplant scenario, okay? Let's hope I can draw this correctly. All right, so we have a thymus. And in the thymus, there are some thymic epithelial cells, the structural cells of the thymus, and they are presenting antigen on MHC class 1, and in some cases MHC class 2, for T cell education, right? This is one very large thymic epithelial cell. And it has on its surface it really has 6 different MHC class 1s, all 3 of moms, all 3 of dads. Mom's ABC, dad's ABC, but I only have enough colors for 4. So I got 4. So it's presenting on all these different MHCs. And different T cells we'll make the T cell match just because it'll make our life easier. There's going to be some T cell that's going to develop and it's going to match that MHC, mom's HLA-A. There's some T cell that's going to match that MHC. There's some T cell that's going to match this MHC. There's some T cell that's going to match this MHC. Ta-da! Those T cells are all going to get some kind of signal in the thymus. They're all going to get to live. They're all going to become T cells. And eventually, they're all going to leave the thymus. Now you can see they're outside the thymus. And eventually, this one somewhere is going to find its MHC presenting a peptide we know these are all CD8 T cells because I'm only drawing MHC class 1. This is going to recognize some, we'll say, infected lung cell. Presenting its peptide of interest on the MHC. And we're going to kill that lung cell and get rid of the virus. Hooray! And this one is going to see a dendritic cell with virus presenting on this MHC type, the blue kind. And hooray, it's going to work. This one is going to present to a dendritic cell that has some bacterial antigen is going to present. We're going to kill. We're going to make a good response against that bacteria. Because remember the importance of dendritic cells in turning on T cells, we need signal 2. In fact, we're going to call this one a dendritic cell 2. It's going to be a dendritic cell with a lung virus. Okay, this is going to be some other dendritic cell with hepatitis virus. And so we're all going to live happily ever after, and we're going to kill all these cells. Yeah? Good here? Now we're going to imagine what might happen if we have to give this person a bone marrow transplant. We're going to make it so that those T cells go away when we do radiation, right? We're gonna have to remake them after we put in some bone marrow. But also, those dendritic cells are all gonna go away when we do our radiation. So we're gonna remake them from our bone marrow. So let's imagine I put in bone marrow, and it has this MHC type. It has 6 copies of that one. What kind of crazy inbred genetics had to happen to this person to have that kind of MHC genetics? I don't know. But that's what it happened. So we're going to put in some bone marrow into this person. That bone marrow is going to result in some T cells getting made. And remember, those T cells are going to just do random VDJ and then try to get educated by what they see in the thymus. And so we're going to get some T cells. And we're going to have one T cell that's going to get educated and say, aha, this is self, because that's what I saw in the thymus. And we're going to have one T cell that gets educated and say, aha, that's self. That's what I saw in the thymus. And one's going to say, aha, this is self. That's what I saw in the thymus. And one's going to say, aha, this is self. This is what I saw in the thymus. But then those initials are going to leave the thymus. Look, they're still there. They're the same ones I didn't erase them before because I was tired of drawing. And they're going to go out and they're going to interact with dendritic cells. Well, we got rid of all the patient's dendritic cells. Where do the dendritic cells come from? Yeah. Yeah, they came from the bone marrow. So those dendritic cells are presenting on the MHC that that person had. Now what happens when these dendritic cells present antigen to the T cells? Yeah, Michael. Why not?
Because the MHCs don't match with anything.
Yeah, we now only have MHCs on our antigen presenting cells that don't match the MHCs of the thymus. And so now we can't do a thing about viruses or whatever pathogens there are. It's not like we didn't make T cells, we made great T cells. But those T cells now cannot respond to the antigen presenting cells that also derive from that bone marrow. Here is the figure from your textbook that shows the same idea. So what you can see is that the donor-derived thymocytes are selected against the MHC types of the thymus, or donor-derived thymocytes are positively selected on recipient allotypes. That means that they're going to be restricted by the recipient MHC, the kind of MHC that you see in the thymus, not the kind of MHC that came from the donor. But the antigen-presenting cells are going to have the donor's type of MHC. So there's going to be no reaction, no response. It's not going to work. The T cells are going to go around forever looking for that self-MHC they learned about in the thymus. And they're not going to ever find those infections, and the person's going to be sick in bed because the T cells didn't work. No adaptive immune response is made. And so that's what happens if the bone marrow doesn't actually match the thymus in terms of that bone marrow transplantation. So it's less about a rejection problem and more about a, can you actually make some T cells and antigen-presenting cells that match one another. So as long as you have some class 1 and class 2 matching with the thymus, then in fact your T cells and your antigen-presenting cells will have some matching MHC, and you'll be able to deal with infection. There are also some times where people try to do super clever things where they actually will do a bone marrow and thymus transplant to try to get around some of this. So this is kind of that extra issue of MHC matches when we think about bone marrow transplantation. I showed you this same slide on Monday. And we saw the number of mismatches in MHC, and this is usually thinking about a solid organ transplant. And the organ's half-life how long that organ is going to last. And you can see that those organs are not always going to last a super huge time. You guys were asking some great questions on Monday about, well, how do we deal with this rejection? How do we make sure this rejection doesn't happen? We saw, minor histocompatibility antigens. And so unless any of us have identical twins, if we need a kidney, there's probably going to be some acute rejection going on because there's going to be some protein that's different. The way that we are able to really make this work is because a medication has been developed that we can give to our patients. And we're going to talk about that medication a little bit, and then some other details regarding transplantation, and then we're going to talk about other medications for allergies and autoimmune diseases and things like that. But you are going to see in this as a theme one problem that you can kind of think about with a lot of these medications. And this medication that I'm going to tell you about revolutionized the transplant field. Basically, there wasn't transplant immunology without this medicine. But there's a big problem with it. And you're going to see similar types of big problems with some of the other medications that I show you. And one of the things that we're going to think about when we think about kind of our next week stuff and how we think about some aspects of immunology for the future is how we try to get over these problems with a lot of these medications. So the big transplant medication is called cyclosporin A. And this figure from your book shows you some details about cyclosporin A. I'm actually going to show you the next 2 slides, and then I'm going to come back to this slide about cyclosporin. So what I want to show you before I tell you how cyclosporin works is how awesome cyclosporin is. So in black, you can see the probability of that kidney or whatever transplant surviving in the patient. So you can see at 1 month, there's sort of a 60-ish% chance. At 3 months, 40-ish. 6 months, a little less, 12, 24, 36. So maybe we gave our patient 3 extra years. For some of our patients, that's a big deal. But that's not, amazing. We have to then, somehow give them a new kidney because their first kidney we gave them got destroyed. Do we have that many kidneys? And so, it's not great. In blue, you can see those survival percentages with cyclosporin. And so what you can notice is that cyclosporin dramatically increases the life of those grafts. And so you can see now, at 36 months, we're up to where we were at actually 1 month in terms of rejection before. Really, transplantation was kind of like a rare, not as much of a thing until this drug, cyclosporin, was there. Here you can see how long patients survived after a heart transplant before and after cyclosporin was introduced. So you can see, heart transplant patients, maybe 50% of them lived. And now you can see it gets almost up to 80% of heart transplant patients live. All because of cyclosporin. And this is living for 5 years. So the reason why I wanted to show you these first is I want you to realize this drug is amazing. This drug has revolutionized a lot of therapy because you're going to see the problem with it as we talk about how it works. And what I've noticed in a bunch of classes over the years is sometimes I tell students about how a drug works, and they'll be like, that drug is the worst. We should let's go burn down the FDA for letting us have such a terrible drug. And I'm like, yeah, it's got some problems, but it's also saved a lot of lives. So it saved a lot of lives, even though we are going to see some issues. So here we can see how cyclosporin A works. On the left, this figure shows sort of what normally happens. And you've actually seen this process earlier in the semester. When a T cell is activated, we will get some signaling that involves calcium release in the cytoplasm. That calcium will bind calcineurin, eventually activating our transcription factor, NFAT, which can go to the nucleus, lead to transcription of IL-2. So this is exactly the calcium pathway that I showed you earlier in the semester. Cyclosporin A, which is shown here as little red dots, and there are some similar types of drugs like tacrolimus, but I'm just going to focus on cyclosporin. Cyclosporin A makes it so that when we have calcium signaling, the calcium still signals, but we can never actually activate calcineurin. So the cell gets full of calcium, but the calcineurin part can never happen. And so the NFAT can never go to the nucleus. You can see NFAT's still chilling in the cytoplasm because the drug cyclosporin is just hanging out here with calcineurin. NFAT never goes to the nucleus. We can never fully activate that T cell. We can never make IL-2. And so what this means is that we're going to keep the T cells off. The T cells are not going to be active. And so the T cells can't go kill the kidney because acute rejection is happening with T cells going to kill the kidney. So we're going to keep those T cells off. We're not going to kill the kidney. Hooray! Sounds great. And you saw the data. Revolutionized transplantation. Saved so many lives. But we've got a problem. Anybody notice the problem as we talked about this? Yeah, Carney.
If the T cells are inactive, although they're not going to kill the kidney, they're also not going to kill the pathogen.
Yeah. When I give this drug, cyclosporin A, it doesn't go through all the T cell. It doesn't go around your body and say, are you the kidney-specific T cell? Are you the kidney-specific T cell? And only inhibit the kidney-specific T cell. It inhibits every T cell in your whole body. It broadly turns off all your T cells. It's broadly immunosuppressive is the term in the field. So now we're basically taking this person's immune system and just moving the whole thing down, suppressing it across the board. We're suppressing all the T cells. So yeah, we're not going to reject the kidney, but now this person could potentially get really sick from some kind of pathogen. Yeah?
Is this similar to what is kind of happening with the bone marrow transplant?
Not exactly, because with a bone marrow transplant, we're actually killing all leukocytes. Here, we're just turning off all T cells.
I'm saying like when we don't have the MHC matching,
Mm-hmm.
like how the T cells
I hadn't really thought about it that way. Maybe. So again, drug is great, saving lots of lives, but it's really not specific. And so what we're going to see in a lot of cases is that some of the drugs I'm going to tell you about, it's like they're kind of like bombs that kill everything instead of just stopping the one bad T cell. We're just going to hit all the immune system. And so this means that the people who are on cyclosporin A, the people who've had a kidney transplant, are extra susceptible to infections. That means that they're extra susceptible. It's going to be really important for them to get, COVID vaccines and wear masks and do all that kind of stuff. And they're going to be super susceptible to stuff. And we're going to have to worry about that. Yeah, Michael.
take cyclosporin A just indefinitely?
And they are basically going to be taking this drug for the rest of their life in order to make sure those T cells never reject their kidney. Yep, Carney.
Is there any way to make the drug more highly selective against?
we'll see a little sort of like movement in that way as I talk about some of the autoimmunity drugs, and we'll see some even further movement when I talk about cancer. But we're still, maybe we used to be this broad. We would like to be this narrow. We're maybe got to here. So it's a little better, but it's not like, it's a general problem in the field. So that's something to be looking at as you look at a lot of these different therapies I tell you about.
So what do those patients do when they have an infection?
They go to their doctor.
Like,
Get a lot of they're on antibiotics frequently. It depends on what the infection is, but they are closely monitored and are under medical care a lot of the time. Is the answer. So one other piece that I want to tell you about transplantation before I tell you about other therapies. And that is sort of this other issue that we have with regard to transplantation. And so this is almost a little bit more of a public healthy thing than a straight up immunology thing, but it's very related to what we're talking about, and it is in fact something that a lot of immunologists have spent a lot of time thinking about. So in yellow, you can see how many people are transplant donors over the years. So you can see about how many people we have who are transplant donors for over different years. In blue, you can see sort of how the numbers of transplants have changed. So you can see we've had a little bit of an increase in donors, but not too much over the years. We've had a bit of an increase in transplants over the years, but not much. But red is the number of people who needed transplants, the number of people who are on a waiting list for transplants. And you can see there's been a huge increase in the number of transplants needed, the number of people on the waiting list. And so what you can notice here is that one of our big problems we have with transplantation is that we don't have enough organs, right? To do transplantation. That's actually one reason why it's really important to make sure your patient doesn't reject your organ, because it's going to be extra hard to get them a second one. And so there are a bunch of thoughts about how we can potentially deal with these problems. One of them is completely a public policy thing. It turns out that different countries have different rules regarding organ donation. So in some places, you are not an organ donor unless you say, I would like to be an organ donor. That's called an opt-in system. You have to do something. If you're lazy, then you're not an organ donor. And if you look at those countries, you can see basically what % of people are organ donors. So some countries like the Netherlands, it's pretty good, 27%. Other countries, it's really low. You can kind of imagine, like, not that many people go through the work of being like, I want to be an organ donor. Most of them, like, go about their day doing other things, and they forget to fill out the paperwork, right? There are other countries where the law is that you are an organ donor unless you say, I do not want to be an organ donor. It's called an opt-out system. There are some countries with opt-out systems. And you can see the % of people who are listed as organ donors in those countries. And what you will notice is that it is incredibly high compared to the opt-in countries. And so one way that we could start to deal with this problem is actually have more donors. Potentially that could be by changing to having things like opt-out policies, where if you're lazy, you don't get around to filling out the paperwork, then by default you're giving your organs instead of by default you're not. And that, again, that's a public policy thing. But that's one way to sort of address this problem. But another way that many people have tried to address this problem is to think about other ways that we can get organs. There are a lot of different ideas out there. Some people have thought about trying to use stem cells to regrow more kidneys So if your kidney is getting damaged, we should take stem cells from you, grow you up a kidney in the lab, and put it back into you. We don't really know how to do that right now, but that's one idea. One idea is that we could use sort of mechanical devices or things like that. Maybe have those try to do the work. One problem with all of these, honestly, is if you can't stop the T cells, then the T cells are just going to go destroy your lab-grown kidney too. And you're going to have to go grow another one in the lab. And so at some point, you'll need to understand the immune response. But then there are other people who have thought a lot about getting organs from different animals and using them as a source for transplant. The most commonly discussed animals for this are pigs, specifically miniature swine. Miniature swine, I was very confused about this in grad school. Megan Sykes worked where I was a grad student, so we actually had to go see the pigs and learn about this. And I was like, oh, miniature swine, and I was imagining those little pigs that live in somebody's purse. Yeah, no, miniature swine are whole big pigs. They are not living in anybody's purse. They're like what you think of as a pig. It turns out that their organs have the same volume as ours. So if you took out a kidney, there would be a hole in your body, and a pig kidney would be the right size for that hole. For a lot of other organisms, the organs are the wrong size. And people have worked on genetically engineering pigs to serve as organ donors. Within the past year, the first pig to human kidney happened. That was actually at the end of 2021. And earlier in 2022, the first pig to human heart occurred, both from genetically engineered pigs. You can see this, is from a couple weeks ago, this article. And so thinking about kind of what the antigens are like and what is the immune response like, and can we actually make organs that can be used for these purposes is another place where there are a bunch of immunologists thinking about stuff. If we look at a lot of the clinical conditions we have been talking about so far, be they transplantation, be they allergy, be they autoimmunity, they all have in common that they're all sort of a situation where you have too much immune response. Goldilocks has too much for all of them. In every case, we want to turn down the immune system to get you back to health. There are separate diseases where you have too little, and we need to turn your immune response up, and we'll talk about those on later days. But all the things we've just been talking about, our immune response is too high, we need to turn it down. And so that's part of the reason why I put the therapies together, because a lot of them are very similar, and a lot of them have overlap. Many of the big treatments for allergies are specifically going towards treating type 1 hypersensitivities. These are two different tables showing ways that one really treats type 1 hypersensitivities. The top of this table on the left is pretty similar to the table on the right, both of which are things we use to treat allergy. This down here is things people are trying to use, but that are not actually happening yet. So one of the big things that we use to deal with a type 1 hypersensitivity is listed in this textbook as hitting mediator action. Really? This is talking about something like an antihistamine. So one thing that you could take to block your allergy is antihistamine. And an antihistamine specifically blocks the histamine receptor. It makes it so the histamine molecule can't interact with its receptor and do its thing. Right? Easy. So that's kind of look at what's going on here. Here is our mast cell. Here is my poor, sad mast cell in my many things I'm allergic to. Got some IgE on its surface. My allergen, like pollen, triggered it. And that mast cell is going to make a whole bunch of stuff. So one thing I can do is I can take an antihistamine, and it will make it so that this histamine doesn't bind to its receptor on my blood vessels, and so I don't get some of these symptoms that are coming from the histamine. Can you see any reason why that might not solve my problem? Yeah, Michael. Yeah. So we blocked the histamine from binding to histamine receptor, but there's still all this other business that the mast cell is making that can be causing me problems. So if we block the histamine, we don't block this stuff, then that kind of sucks, right? If you think about newer allergy medicines, you end up seeing things like this. Can I make it big? Apparently I...
Substance. Flonase controls 6, so you are greater than your allergies. Flonase, 6 is greater than 1.
There we go.
Flonase allergy relief nasal spray outperforms the #1 non-drowsy allergy pill. Most allergy pills only control one inflammatory substance. Flonase controls 6, so you are greater than your allergies. Flonase, 6 is greater than 1. This changes everything.
What do you think Flonase does? Yeah, blocks more of them. Doesn't block all of them, but it blocks more of them. If you look at the Flonase package, it says, it tries to use some fancy terms for you to tell you what it blocks. It blocks histamines, and it blocks cytokines, and it blocks chemokines, and I don't remember what the other ones were. But I just remember laughing, looking at a Flonase package, being like, oh, those are your fancy 6, huh? So it's blocking more things, but it's actually still not going to be able to block every single thing. And the other problem is, like, if I take my Flonase, my mast cells I've already got this IgE. I've already got mast cells that are coated with IgE. My mast cells are already ready to go. It's kind of like, stopping the train right at the end of the train, but not actually stopping the whole long process. Similarly, so you can see, there are some things that block some of the other molecules as well. Sometimes what people try to do is say, well, what if we stop the IgE from binding the mast cell? So some people are treated with anti-IgE antibodies. Those might bind up all the IgE, so the IgE can't get to the mast cell, which can be pretty good. But again, if you went to the doctor because you have allergies, you probably have at least some mast cells that already have IgE on them. This isn't doing anything for those mast cells. But it can help you stop making more mast cells that are going to be coated. So that could be helpful. It's not going to be, everything. We can also use things like epinephrine, aka an EpiPen. Epinephrine is, also known as adrenaline. It's a major hormone that your body makes. And it happens to actually also mess with mast cell signaling to try to stop mast cells from degranulating. So you can basically stop mast cells from degranulating if you, are in the midst of a severe bee sting response and you're going to die. stopping your mast cells right now could be really helpful. It's not going to protect you in the future, but it's going to stop your mast cells and make you not die right now. Similarly, we can use some other molecules to sort of chill out aspects of the immune system. These are other hormones. Most frequently, they're versions of cortisone or a corticosteroid. You can see cortisone here. Most of the time, what we tend to treat patients with is not straight cortisone. We actually use prednisone. You may have heard of prednisone. You can see the structure of prednisone here. It's a steroid. It's basically a corticosteroid. It can diffuse through the cell membrane and bind to a protein in the cell, and then directly change transcription. And that's how all of these corticosteroids work. And when that happens, we will decrease a lot of cytokine production, will decrease production of some other mediators like nitric oxide, will decrease making a lot of the lipids, will decrease the adhesion molecules. We actually will induce some lymphocyte apoptosis. It's not shown here, but we'll actually suppress lymphocytes. Basically, if we treat with that steroid, prednisone, we're just sort of broadly like putting a blanket on your immune system. We're like taking everything and being like, and pushing it all down. Yay, you stop having the rash or the whatever allergic response you have. Hooray! What's the problem? Yeah. Yeah, your whole immune system got non-specifically reduced. Generally, when you're on something like prednisone, you're on it for a short period of time until the allergic reaction stops. But again, we're not really targeting just the cell that is causing the allergy. We're just calming everything down. I will also just quickly note that you're seeing here prednisone, which is a corticosteroid, kind of doing this general immunosuppression. There are a whole bunch of different types of steroids that are sort of related here, particularly to cortisone or these corticosteroids. One of them is called cortisol. Anybody know anything about cortisol? When might you make cortisol? Yeah. Yeah, when you're feeling super stressed out, you make a hormone that basically is very similar to this. It's gonna down your immune system. How many of you often feel like you get really sick right after the semester ends? Because you were really stressed out, your body made a lot of stress hormones, you did this broad immunosuppression from that cortisol, and in fact, maybe you were a bit more susceptible to infection. So we use this with prednisone and corticosteroids, but you can also see that it plays into some other physiologic things you might be aware of. One other thing that is frequently used for treating allergies is listed. So here you can see the corticosteroids, which have general anti-inflammatory responses, is something known as desensitization therapy. You might have heard of getting allergy shots. Allergy shots is desensitization therapy. And so basically what we are trying to do with desensitization therapy is give you a whole bunch of shots of a really low dose of the thing you're allergic to induce tolerance. Ideally, we're trying to exhaust those T cells, deplete those T cells, push you towards tolerance of that allergen before you, instead of your allergic response. So we're trying to deviate that response either to a Treg or to some other helper T cell subsets. Sometimes this can come from feeding you your antigen and trying to get you to push to oral tolerance your allergen. Sometimes it can be through large numbers of allergy shots. You can imagine there's some danger in this one. This is done usually under the care of a physician in your physician's office. I know someone who had a bunch of allergy shots, and at first they always had to go to their doctor's office, and then they got to do them at home. But when they did them at home, they got an EpiPen so that they could cure themselves if ever it went wrong. For some people, it works pretty well. For other people, it does not. There actually has been some pretty good use for people with really, really severe peanut allergies. Like, there are people who have kids who have peanut allergies so severe that if like someone else in their elementary classroom opens a bag of peanuts, they're gonna have a response. Desensitization can knock that response down so that like they're never gonna eat peanut butter sandwiches, but they can be in a room with someone else who has peanuts and not die. And so we can get that response down in a lot of cases, though it may not actually go away completely through desensitization. Yeah?
Wouldn't oral tolerance be a little bit more risky since you're eating what's making the anaphylactic thing? So, and you'd still have it in your system? So
Well,
wouldn't that
well, hopefully, because the GI tract is an immunoregulatory environment, you're going to push a lot of Tregs, is the idea. But it's always risky. So this is sort of that other idea. Again, you can see like it's good because at least we are now trying to target the specific cells. We're not just turning down the whole immune system, but it's still got some problems. We see a lot of similarities when we think about treatments for autoimmunity. So this table talks about different things that are used to treat autoimmunity. One of the things that is not shown on this table, but that it is used in some patients, is prednisone, or those same corticosteroids that we saw for allergies. One thing that is shown on this table as an autoimmunity treatment is cyclosporin A. For the same reason. We're going to turn the T cells down so they can't give us an autoimmune response. But we're going to have those same problems of now we are basically going to have that person on cyclosporin for the rest of their life. And they are going to have increased susceptibility to infection. So good news, this patient is going to be protected from having their autoimmune symptoms, but they're probably going to have some other things as well. And as I said, this is a super broad thing. And in these therapies, this breadth is a little bit of our issue. As we'll talk about next week, when we think about cancer, one of the problems with cancer is sometimes the breadth of the therapy as well. It's hard to target just the one cell. And so in all of these cases, what we really want in our pharmaceutical is specificity. We really want to figure out a way to make our drug specific to the cell we want to act against. Have you heard the word specificity ever this semester? Immunologists love the word specificity. Specificity is like the defining feature of the adaptive immune system. Our whole system is about specificity. So maybe we can think about ways to use pieces of the immune system that are super good at specificity in order to deal with some of these issues. And so one of the things that we use, I'm going to be talking about it now for autoimmune diseases, but it's used for a lot of types of diseases besides autoimmune diseases too, is that we now will use biologics as therapies. And specifically, in this case, we will treat people with antibodies that were made in the lab, monoclonal antibodies that can specifically block something. And we'll talk about what the something is going forward. Originally, people were using antibodies that came from a mouse, or came from mouse cells. But they worried about serum sickness, that type 3 hypersensitivity response I told you about, using foreign antibodies. And so pharmaceutical companies have figured out ways to make the antibodies in a lab that are more and more human antibody and less mouse antibody. So sometimes people made antibodies that were just the variable region from mice and the rest of it from human. People have figured out ways to have just the CDR3, the 3 CDR loops from mice, everything else from human. People have also figured out ways to make fully human antibodies in a mouse. It's pretty cool. Because even here, there's these little strips of foreign protein that you could make an immune response to. Here, there's nothing foreign. If you've ever seen a drug whose like actual drug name ends in MAB, blah, 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 MAB. That means that drug is a monoclonal antibody. If it ends in OMAB, it's a mouse monoclonal antibody. If it's ZMAB, it's a chimeric. If it's UMAB, it's fully human. If it's Zixu, I don't know what that is. If it's Zumab, anyway, you can see sort of this nomenclature. When you now see drug ads on TV or like in a magazine or something, look for the drug name. You will be shocked at how much of the time it's some kind of MAB. You can see a list of some of them here that are in use for different patients. You can actually see that some of them are used for transplantation, some of them are used for cancers, which we'll talk about when we talk about cancer. Some of them are used for different autoimmune diseases. You can also see that most of them have been approved relatively recently. And if we think about the pharmaceutical industry, this is 2009 to 2014, which was like the beginning of the monoclonal antibody revolution in pharma. You can see that then it increased by 63%. It would be dramatically higher now. This is the direction that pharma is going. This is how everyone's making all sorts of pharma drugs. So if you decide, I'm never going to think about immunology ever again, but I'm going to go make money in a pharma industry, probably if you're going to make a lot of money, you're going to be making it on some kind of monoclonal antibody, like one of the ones I'm telling you about. These antibodies can do a few different things. So some of them are like the ones shown here. This includes the first one that was really ever used for treating patients, rituximab. You can see that both of the ones listed here are listed for rheumatoid arthritis, although they're actually used relatively broadly now, especially in rituximab. Rituximab is an antibody against CD20. CD20 is on the surface of mature B cells. And so what we do is we give our patient antibodies against CD20, antibodies against B cells, and the B cells get killed. So with rituximab, we're basically getting rid of mature B cells in our patient. If our patient has an autoimmune disease where they're making antibodies that are causing them problems, you kill the B cells, you stop making the antibodies. But what's the problem? Yeah. You killed all the B cells. Cyclosporin actually inhibits B cells and T cells. So now you're a little more specific. You're just killing only the B cells instead of hurting both. And in fact, you're only killing the mature B cells, so you still get the naive ones. But not great. You can see there's a similar thing where you can actually kill all the CD4s with a CD4 antibody. So that's one thing you can do. It's a little more specific, but not great. One of the other big molecules that's very similar to this, it is known as abatacept. So officially it is not an antibody because there is some other engineering. You might have heard of the drug Orencia. You might have also seen the Nobel Prize, which was given for this a couple of years ago. You can see that this is used for rheumatoid arthritis, lupus, inflammatory bowel disease. This one's used a lot. And this is an antibody-like molecule. We've actually fused our own thing onto an FC. So that's why it's not officially an antibody, but it's basically an antibody. And here you can see that molecule here. It is CTLA-4 with an FC portion. So it's sort of like CTLA-4 made into an antibody. Here you can see that CTLA-4 with the antibody. We talked about CTLA-4 before. Remember that CTLA-4 is on the surface of some T cells in order to turn them off. And the way that that happens is that CTLA-4 binds to B7 really strongly. So in fact, that B7 can't be given a signal to anymore. If you treat a patient with this CTLA-4 protein, that CTLA-4 protein is going to bind to all the B7. And now our T cells can't get signal 2. So basically, all the B7 is going to be blocked by this protein that you've delivered, this CTLA-4 protein, this drug. And so none of your T cells can get signal 2. Great news. We don't turn on T cells now. We can maybe help our patient with their autoimmune disease. Little better, because again, there still are some other kinds of signal 2s that we didn't really talk about, but there are a few other ones, but they're like minor. So there is maybe a way you could still deal with pathogens, but the main famous way of making T cells work doesn't work. So like maybe you do get a few T cells that still can work, but it's still not like awesome. Still not super specific, as you were saying. A little better, but not great, but better. And again, Nobel Prize, like amazing drug, yeah.
Doesn't this also have a cascade effect because these cells can't be fully activated?
It does, yep. But again, it kind of is like, well, but maybe you could use some kind of off way of turning on the T cell, and that T cell could turn on the B cell. But you're right, the cascade, absolutely. And so this could stop the T cells, this could stop your B cells from making autoantibodies, and also immunosuppress you. The other kind of big thing that we see are cytokine-based therapies. And so there are a few cases where people actually treat you with cytokines. Those are actually quite rare, and none of those work super well unless you're in really bad shape. I showed Dallas Buyers Club in my DSEM, and at one point in the movie Dallas Buyers Club, the HIV patient tries to inject himself with interferon beta to cure himself or to help his HIV, and then he immediately goes into a coma in the middle of an airport because the interferon is so strong. So usually we don't actually just inject you with cytokines. That's pretty rare. But more frequently, we use antibodies against cytokines. And so we either will use an antibody that will bind up all the cytokines, or an antibody that will bind to the receptor. Thanks, Siri. An antibody that will actually make it so the cytokine can't get to the receptor, a blocking antibody. And the antibodies bind to the cytokine or the receptor and block the cytokine from signaling. So you can see some of a short list of this. There is a huge list of these. The most famous of them will work against TNF-alpha, which were the first ones that were described, although now they have, the TNF-alphas were kind of the ones for a long time. So if you've heard of Enbrel or Humira, which are the the trade names, those are both anti-TNFs. And again, for a lot of patients with a lot of autoimmune diseases, they were revolutionary. We've now found a lot more of sort of IL-7s and IL-23s and things like that that seems to be helping a lot of patients. But these drugs are revolutionary for the patient. So these are just showing you with a rheumatoid arthritis patient kind of what happens to that patient in terms of all of their symptoms compared to placebo if those patients have an anti-TNF antibody. And so, like I said, Remicade or Humira, Enbrel, blocking TNF has basically changed the life of autoimmune patients. My dad has an autoimmune disease. When I learned about these actually in my first year of grad school. It wasn't approved yet for his autoimmune disease, but I got him on it because I learned about it in grad school. And it took something that he basically had suffered a lot with his whole life and completely made it gone. These are revolutionary drugs. This is just showing you that we can hit pretty much all parts of T-cell activation. The good news about things like these is that now, instead of stopping the whole T-cell, you're only hitting one cytokine, so only a little, like a smaller part of the response. So if a T-cell makes 3 cytokines, you're only turning off one of them. So you're not as broad, but you're still turning off all of the TNF, all of the T-cell responses. And so in the end, you get something that is like this. It's this one. No. It's this one.
I'm Phil Mickelson, pro golfer. My psoriatic arthritis caused joint pain.
Just like my moderate to severe rheumatoid arthritis.
And I was worried about
Joint damage. My doctor said joint pain from RA can be a sign of existing joint damage that could only get worse. He prescribed Enbrel to help relieve pain and help stop further damage.
Enbrel may lower your ability to fight infections. Serious, sometimes fatal events, including infections, tuberculosis, lymphoma, other cancers, nervous system and blood disorders, and allergic reactions have occurred. Tell your doctor if you've been someplace where fungal infections are common, or if you're prone to infections, have cuts or sores, have had hepatitis B, have been treated for heart failure, or if you have persistent fever, bruising, bleeding, or paleness. Don't start Enbrel if you have an infection like the flu.
joint pain and damage.
Can go side by side.
Ask how Enbrel can help relieve joint pain and help stop joint damage.
Enbrel, the #1 rheumatologist prescribed biologic.
This drug is amazing for many, many patients, but you also just heard half of that commercial about all of the potential side effects and all of the things that can go wrong. And again, we've made this a little narrower than some of the early ones, and we still have this huge list of potential issues. We have changed all of these patients' lives. Again, all of these drugs are revolutionary. People are making money at the pharma companies like swimming in their Scrooge McDuck dollar bills from these drugs. And again, there are many people whose lives are dramatically improved. But you can also see there's also a lot of room for us to think about the immunology of all of these processes and try to improve. I'll see you guys in the lab tomorrow. The lab handout is posted. Yeah, that's that.